Hey y'all, what's up, baby penguins? So we are live, so we can play some Unit 2 review games. So I discovered last time that in order to actually be able to play the games, I need someone to actually enter the game. So I am going to awkwardly sit here while I wait for someone to join the live and also join the game. So I figure I give about 5 minutes for people to join the live and join the game, just because it may take a couple moments for people to get the notification that we're live. So just hang in there just a moment while we wait about 5 minutes to see if anyone comes in, see if anybody wants to join our review game with us. You know, the code is 673901. Go ahead and join the game. I discovered last time in order to play the game, I actually have to have someone in the game. So if anyone wants to come and join our game, come and join our review session, that would be amazing. And then if you also want to type in the code into as a question, then I can pin it so that if someone joins later, they'll be able to join the game. So anybody who's in the room, if you would just, happily go to joinmyquiz.com and type in that code, even if you're not actually going to play the game, that way I can play the game live. Yes, I got someone. Yay. So I figured that I would wait just a couple moments, see if anyone else wants to join the game. If you can do me a favor and type the code into the little question box, then I can pin it so that anybody who joins after we get started, then I'll be able to, they'll be able to join the game. Yay! Somebody, no, that's just asking in the regular chat box. I need it to be in the question. I don't know how to pin it unless you all know something that I don't know. I think whenever I click on your name, all it does is like tell me that I can do stuff too, like block you and stuff. That would be nice. So, give it just a moment, see if anyone else wants to join before we get going. Hope y'all are doing good tonight, or I guess it's daytime, it's like 4 p.m. So, how's everyone doing? Hey, someone asked in the question box. Awesome, you're amazing. Thank you. There we go. Now I got it pinned. Thank you so much. That way it'll be up so if anyone joins the game a little later, they'll be able to actually get into the game. And again, I said I'd give it just a couple moments so that anybody who needs to join, like maybe they didn't know that I've gone live, they're waiting for the notification, all that good fun stuff. So I'm just waiting just a couple moments. Where y'all coming from? Y'all excited to review Unit 2? Like, I know I am. I just feel like it's kind of early to review, but like in order for us to have enough time to kind of get through each of the units, which each of the weekends, we kind of have to start this early to review. But I did start going, all of my review questions on my Instagram, so that's always good. Okay. Well, I don't see anyone else joining, so I'm going to press the start button. I know it's a little earlier than I said, but that's okay. So let's start. And it should show up on your screen instead of my screen. I have to show you this way, but this way you can at least see it on your side. So we're describing the differences between the rough ER and smooth ER. Everybody's in. Okay. Okay, so describe the difference between rough ER and smooth ER. Rough ER we know is going to, it's shifted. Sorry, y'all. So we know it's going to compartmentalize versus our smooth ER is in charge of our detoxification or lipid synthesis. We also see that our rough ER is going to have ribosomes on it, and it's going to allow for us to make membrane proteins as well as proteins for secretion. And then the smooth ER we also know is going to be responsible for storing calcium. So these were just some of the functions that we had. Next question. There was a way I saw it last time that allowed me to. So which organelle is a membrane-bound structure that functions in correct folding, chemical modification of proteins? Oh, you guys are all in. Okay, so which is the membrane-bound structure that functions in chemical forming and the folding of it? So that would be our Golgi complex. The Golgi complex is going to be right after we have our rough ER. And so we'll see that we can modify those different proteins. The lysosome is going to be responsible for digestion, so we can break down materials. Mitochondria is where we're going to see part of cellular respiration will have the oxidative phosphorylation, as well as we're going to have the Krebs cycle. The chloroplast is where we'll see photosynthesis, so you'll see the light reactions as well as the Calvin cycle. There's got to be a way that I can see the timer at the same time as seeing the, let's call it, the question. Okay. Next question. There it is. That's granny. Which membrane enclosed sacs contain hydrolytic enzymes? How are you already done? Y'all are just really fast. So this is exactly what we just looked at last second ago. So hydrolytic enzymes means that it's going through hydrolysis. Hydrolysis means that we're going to be breaking down some type of polymer into monomers. And so the lysosome is responsible for breaking those bonds using those hydrolytic enzymes. Versus we saw last question, the Golgi complex is going to be responsible for modification of those proteins that are made in the rough ER. Mitochondrial cell respiration, and then chloroplast we said was photosynthesis. So great job, y'all. If y'all have questions along the way, just let me know. Which one is responsible for storage and release of macromolecules, cellular waste products, in addition aids in turgor? So y'all already got the answer in. I think we only have 2 people in the room versus 3 in the game, so I know that there's no one in the game waiting. So what we see here, this is going to be looking at your vacuole. So we have a central vacuole in plants that's going to be responsible for that turgor pressure. It absorbs all that water and then provides that the pressure against the cell wall. That's why we talk about the 3 different kind of, if there's too much water in it, it's got turgid. If it's in an isotonic environment, then it's going to be kind of normal and flaccid. And then it's in a hypertonic environment, then it's going to be plasmolysis, where the plasma membrane pulls apart. We also have food vacuoles. Those food vacuoles are responsible for like when you engulf food through phagocytosis, that will then bind with the lysosome for digestion. And then we also have contractile vacuoles that are going to kind of pump to push out excess water in a lot of our like protozoa that live in a hypotonic environment. And we already mentioned lysosome is for digestion, mitochondria, cell respiration. Oh, I'm in a closet, so somebody just follow me. And then endoplasmic reticulum, the rough and smooth that we just mentioned a little while ago. Looking at membrane proteins, looking at for the rough ER, and then storing calcium detoxification for the smooth ER. Which of follows the location for the light-dependent reactions? Hey, is Miss White in the room? Hey, Miss White. Hey, so y'all are all done with your answers. So which of the following is the light dependent? So we know the light dependent, of course, requires light. So that is part of photosynthesis. So that limits it down to the 2 that are in the chloroplast. So we have the thylakoid membrane, which is where we're going to have those light reactions. That's where the chlorophyll is. It's going to absorb that light to allow for those light reactions. Versus the stroma is going to be like the jelly substance. And that's where we're going to find the Calvin cycle within photosynthesis. The matrix is where we're going to see the Krebs cycle. And the cristae is where we're going to see the electron transport chain. That's going to be responsible for the electron transport chain, which is part of oxidative phosphorylation. And nice job. Y'all are doing good. Which of the following is the location for the Krebs cycle? If you were just listening, I just told you the answer. We got one answer in, which is fine. Miss White, this is every other week that I do the TikToks. I do the YouTube lives on the opposite weeks. So it's kind of every other Sunday that I go live on TikTok, and then the opposite ones are on YouTube. Okay, so our last person's come in. So as I said before, Krebs cycle is going to take place in the matrix, which is a part of the mitochondria. So if you kind of think about the mitochondria, they have kind of the outer ring, or not, it's not the outer ring, the outer membrane, and then there's that inner membrane that's kind of highly folded, which is where we're going to see on the inside, that's where the matrix is. So that's where you'll have the Krebs cycle. And then in the cristae is where we'll see the electron transport chain. So that's where we're going to see those electrons that are from NADH and FADH2 going down in energy, which is going to pump a proton into that intermembrane space. So it'll be between the, kind of that matrix and like the cristae is kind of outside in the inner membrane. Anyways, so we said the light reactions were in the thylakoid, and then your light dependent reactions were in the stroma. Okay, so let's see what our next one is. Next question. Which the following location for ATP synthesis and cellular respiration? Ooh, we got a new person. Yay! We're up to 4 players. Woo-hoo. And I like this because it goes onto y'all's devices instead of you trying to read it off of my screen, which is good because, you know, my screen's sometimes small, but sometimes not. Five, four, three, two, one. All right. So what we see here is we're looking at ATP synthesis and cell respiration. So we tried before that the cristae is where the electron transport chain is. And after those electrons move down in energy, what they're doing is they're pumping a proton across the membrane into the IM space or the intermembrane space. That potential energy then flows down its concentration gradient through ATP synthase that's also located on that cristae. So that chemiosmosis is what generates ATP in cell respiration. You generate ATP in the light reactions, which is part of photosynthesis. But number one, the question says cell respiration. And number two, that ATP is completely 100% used for the Krebs cycle. I'm sorry, for the Calvin cycle in the stroma. So we're not really going to look at that ATP in terms of for cellular power. And as I said before, matrix is where we have our Krebs cycle. So, which of all is location for ATP synthesis and photosynthesis? So if you were literally just listening to me 2 seconds ago, then you know the answer. I probably need to stop explaining all the other answer choices just in case. But I like to try to help as I go along. There's only 2 people in the room, and 2 people have answered. So who knows? We will see. Okay, so if we're looking for ATP synthesis and photosynthesis, then that's when we're looking at the thylakoid membrane. That's where we're going to see the light reactions. So in the light reactions, you're going to see that the chlorophyll is going to absorb the solar radiation, those photons, which will then allow the reaction center complex to kind of be sent up to the primary electron acceptor. And then it goes down an electron transport chain. When it does this, the protons are again pumped. They're pumped into the thylakoid, and then they fall back down their concentration gradient through another ATP synthase. And so we can synthesize ATP in the thylakoid membrane. We already said before, ATP is made here in the cristae, but for cell respiration, stroma is our Calvin cycle, and the matrix is our Krebs cycle. So relationship between cell size and surface area volume ratios. There's I think content that's in IB bio that isn't an AP bio. I believe y'all still learn about the plants. And I believe there's also a difference in the sequence, but I don't think there's huge differences between the two. I think the difference between IB and AP is how you're assessed. I think you have more of analytical stuff, not really analytical stuff, but you do more than we do, I think. Okay, so describing the relationship, we know that as a cell gets larger and increases in size, the surface area to volume ratio is going to decrease. The reason why this happens is because of the fact that the surface area increases by a square, and then the volume is going to increase by a cube. And so the inside of it increases larger than the outside of it when the cell gets larger. And so the surface area to volume ratio actually gets smaller because of the fact that ratio of the denominator is going to increase faster than the numerator. I'm thinking about joining AP Bio next year. I don't think the course is too hard, but you'd probably want to ask your other classmates that are taking it. As a teacher of it, I don't find it to be very hard. It's challenging. It has its hard moments, but overall as a course, it's not like too hard or too bad. Characteristics of a membrane-bound protein. And the disadvantage of Quizizz is that I have to actually read all the questions and come up with the answers too, since it doesn't tell me the answer on this screen. Ha ha ha. Yeah, so if we're kind of come up with the characteristics of membrane-bound protein, we have to think about what the membrane actually kind of is, right? So the exterior of a membrane, where the hydrophilic heads are, is going to be hydrophilic, versus the core of the membrane, where those fatty acid tails are, is hydrophobic. So in order for a protein to sit in the membrane and be membrane-bound, it has to have hydrophilic R groups that are on the exterior of the cell, and then hydrophobic R groups that are on the interior of the membrane, because then they kind of are able to match the similar polarity in those regions. Nice job, everybody. Identify which of the following will easily pass through the membrane. I think I increased some of the times on some of these just to ensure that there was enough time, since we were, online, and you know how sometimes there's a lag. So I think that I may have increased the timing when I did this when I was ready for the fall semester final exams. I did the live streams, which I don't think we got through as many of the games that time as I wanted to. That's okay. Okay, so if we're trying to think of what will pass through the membrane, we want something that's going to be able to pass through that hydrophobic core, or the part where the fatty acids are. So it needs to be able to pass through hydrophobic. So we know small and nonpolar items can pass through. Water is polar, although it is small, it is polar. So it can pass through the membrane, but not easily. So you might see just a couple molecules of water passing on their own. For the most part, water is going to need an aquaporin to pass through. Protein hormones are going to also be polar. So since they're polar, they also don't easily pass through the membrane. And then charged ions, again, there's a charge on it, so those are polar and they're unable to pass through the membrane. So we're going to see that a steroid hormone will easily pass through the membrane. That's when we see in our cell communication, we'll see that steroid hormones are going to have intracellular receptors, because they're going to bind to a receptor on the inside, since that steroid hormone passes straight through the membrane. There is a FRQ question on that about estrogen from a couple years ago, talking about there was a receptor that would bind to the estrogen, and that's how they would working with, it was a drug, not a drug, it was a drug research question from a couple years ago. Which of the following will bind to a membrane protein but will not enter the cell? So we're looking for what is the ligand that will bind to a receptor on the membrane. Okay, y'all. So we already talked about that water is going to pass through, but it's just not really easily. Charged ions also will go through the membrane by using transport proteins, usually a channel protein or a carrier protein. So if we're thinking about a ligand as something that's going to bind to a receptor but not enter, that's going to be your protein hormones because then they're going to bind to membrane receptor, and then that's going to allow for us to see that they're going to cause cell transduction pathway, because if we see the reception, then we go through transduction of some type of phosphorylation cascade that leads to some type of response. Nice job, y'all. Okay, which of the following requires an input of energy for membrane transport? And again, if y'all have any questions along the way, you're more than welcome to type into the chat, and I can answer those questions as we go too. Has this been helpful so far? Is this a resource we want to continue each weekend? Okay. So if we're trying to figure out what's going to require some type of energy, right? We need to think about something that is active transport, right? Something that's going to go against a gradient, something that is going to have to be pumped in some way, right? So a hypertonic cell means that there's going to be a high solute concentration and a low water concentration. So water moving into that is going to go with its concentration gradient. Charged ion moving with concentration gradient, of course, is going with the gradient. Water moving out of a hypotonic cell. Hypotonic means that it has a high amount of water and a low amount of solute. So moving out of the cell is going to be down its concentration gradient. So the only thing that moves against the gradient is, of course, this first one. And it actually says against the gradient in the answer choice. Nice job, y'all. Difference between endocytosis and exocytosis. Okay, so endocytosis makes us think of something that's entering versus exocytosis makes us think of something that's exiting. Okay, enter is endocytosis, exit is exocytosis. And so we're going to see that endocytosis is going to be phagocytosis, which is cellular eating, and penocytosis, which is cellular drinking to bring materials into our cell. Versus exocytosis has to do with when we talk about a protein that's been secreted by our rough ER, it'll go through exocytosis to kind of secrete it out of that cell. So the answer should be, I think, this one. I got distracted because there was something in the chat and I was reading it. So I just have to read the answer choices. Sorry, y'all. Okay, so we got two more questions. Which of them correctly describes the movement of water? So you have to figure out which of these cells are going to have their movement of water correctly described. Okay, so if we're looking for the correct description for the movement of water, we want to see which of these is talking about a hypotonic solution losing water to a hypertonic solution. And so it says that a cell that has 1 molar solution will gain water from the outside environment of 0.5 molar. That sounds kind of weird, but when you think about the molarity, you're looking at the concentration of the solute. So you have a 1 molar solution, right? So there's a higher concentration of the solute with less water versus a 0.5 molar concentration is going to have a lower solute and then more free water because we're looking at where that free water is moving. So free water is going to move from where there's a higher concentration to where there's a lower concentration of free water. So it should be that third answer choice. And this should be our last question, and then we'll pick a different game for y'all to play. So why is eukaryotic cell able to be larger than a prokaryotic cell? I think the other games may not have been reset with our timing, so we probably won't have as long of a lag in the other games. So sorry, y'all. I know it seems like quite a little long lag sometimes. Some of the questions are longer than others, so I think it's okay. Okay, so when we think about eukaryotic cell versus prokaryotic cell, eukaryotic cell does have a nucleus, and it also has membrane-bound organelles. These membrane-bound organelles are going to provide compartmentalization to the cell. You know, think about that the mitochondria is where we see cell respiration. The RFER is where we're going to see, you know, synthesis of proteins. The Golgi is going to, you know, modify. We look at that the lysosome is for digestion. And so all these organelles have their own specific functions. So it allows for that compartmentalization for those processes. So that allows the cell to be larger because you have all these little compartments that can do different things. So great job, y'all. And that should be the end of the game. Yes, we want to exit. Okay. So we need to figure out what our next game is going to be. So I'll let you write it in the chat which one you want to play. So we have plasma membrane, we have diffusion and osmosis, and we have organelles. So do you want to play plasma membrane, diffusion, osmosis, and organelles? Those are our 3 options that we have of games that I have pulled up ready for us to play that are Unit 2. So again, can you type in the chat which one you want to play next? So we have plasma membrane, diffusion and osmosis, or organelles. Which game do you want to play next? Okay, I see a vote for diffusion and osmosis, and there's literally only 2 people in the room. So I'm not really sure if there's another vote. So we're going to go with diffusion and osmosis. So continue. I think I've got all the settings set up so you can do the other part. Okay. Name factory. Let's get that. Okay. And let me get rid of this so that we can have the game down. Okay. So our new code is 373530. I'll give just a moment for whoever is joining us to join the game. So 373530. And if you don't mind, someone please type it into the little question box so I can pin it for anybody who might join the game later and wants to, or join the live later and wants to join the game. So 373530 is our code, and we've got a person in. I kind of like the random generating for the names just to kind of keep us all safe, you know? So if you don't mind typing that into the little question box, so we'll be able to have that open for anybody in the future that wants to join. So 373530, if you could type that. Okay, awesome. Someone already did that for me. Thank you. Post. Okay. So there's only 1 person in the room and 2 people in the game, so we're going to go ahead and press start. And hopefully they have less timers on them. Whoa. You're a farmer growing crops. The concentration of your potatoes is 0.35 molar, and you're advised to water your plants with 0.35 saline solution to provide nutrients. What happens to your plant cells? Oh, everybody's already voted. Sorry. So what's going to happen is that our cells are become flaccid. because it's an isotonic solution. You notice that the 0.35 and a 0.35, so they are the same concentration. So water is moving equally inside and outside the cell. And if you remember in our last game, we talked about that the central vacuole is responsible for that turgid pressure. It's responsible for holding that water to apply the pressure to the cell wall. If we don't have that pressure because we're in an isotonic solution, it makes the cell flaccid. Normal would actually be if it was in a hypotonic solution. Oops, we got you on that one. Flaccid means limp. So you are a doctor. A patient comes in dehydrated. What type of solution would be best to place into their IV? So those basically what you put in the needle that goes into their body. Okay, so it looks like we have all of our votes in. So a hypotonic solution means that it's going to have more water. So that's going to cause the water to rush into the cells, which will cause those cells to burst, which is a problem, right? A hypertonic solution is going to cause the water to leave the cells and cause the cells to shrivel. So we want there to be an isotonic solution so that we can kind of make sure that there's more fluid in the body without causing there to be harm to the actual cells. Because a red blood cell or a human cell, animal cell, does not have a cell wall. And so it needs to be in an isotonic solution. Great job, y'all. Which following describes an appropriate water movement pathway? So which way does water move? Okay, so here we're going to see that water moves, of course, from a high water concentration to a low water concentration. Hypotonic solutions means that there's a high amount of water and a low amount of solute. Hypertonic means that it's a high amount of solute and a low amount of water. So water is going to move from a hypotonic solution to a hypertonic solution. Oh, we got you on that one. Sorry, y'all. Does it tell you immediately whether the answer is right or wrong, or does it wait until after everybody times out? Difference between a molecule transport to facilitated versus simple diffusion. And yeah, you can type your answers into the chat. That's perfectly fine. So when we look at facilitated versus simple diffusion is going to allow for a material to pass directly through the plasma membrane. It doesn't require any type of protein. It'll just pass it straight through. Both of these are going to types of passive transport in which they're going down concentration gradients. Facilitated diffusion is going to require some type of protein. So either has a channel protein or a carrier protein. Channel protein is just going to provide a hydrophilic region for it to pass straight through. Versus a carrier protein is actually going to bind, have a conformational shape change to move the material across that membrane to come out on the other side. So the answer that we should have had here was facilitated diffusion involves a polar solute transported through a membrane protein. Due to the fact that the solute is polar, is not going to be easily able to pass through that nonpolar core of the membrane, so it should have been that one. Nice job, y'all. So which of the following describes the difference between passive and active transport? Oh, I just realized y'all answered. So when we look at passive transport versus active transport, passive means that it's going to be moving without some type of energy resource because it's going to go down its concentration gradient. And then active transport is going to require some type of energy because it's going to be moving against this gradient. And I have not actually read these answer choices to find which one was the correct answer. So here we see, no. So active transport moving against the concentration gradient, ATP. So that should have been the second answer that we had because I was drinking water instead of looking at the question. Oops, sorry, y'all. Okay, so which of the following describes the difference between channel and carrier proteins? And I just mentioned this a couple questions ago. Okay, so we said that channel protein was going to provide a channel through the membrane to allow for the materials to pass straight through. The problem with this answer choice is that it's a non-polar channel. If the solute was non-polar, then it would be able to pass through that membrane without the use of a resource. You're not helping me, y'all. Go away button. Go away button. Whatever. Versus we want the correct answer, which is talking about that it's going to bind to it and then move it across the membrane. Are you really not going to go away, machine? Oh, there it is. Okay. Next question. Describe what would happen if you place an animal cell in a 1 molar solution. And this is based off of an FRQ that we had a couple of years ago. So if you don't really you kind of need a little bit of background noise for that one. So hopefully you get the answer right. But that's okay. Okay, so here we see that we're in a 1 molar solution. 1 molar solution is more concentrated than the animal cell, which means that it's placed in a hypertonic solution. Since the cell is in a hypertonic solution, the water is going to move out of the cell and into the solution. So that's going to cause the cell to shrivel. Good job. Y'all got that. So what would happen if you put a plant cell in a 0.0 molar solution? Oh, you're welcome, Vicky. I'm glad that I can be helpful. All of my materials are always free to y'all, so anything you need, I'm always helpful and however I can be. If you don't know about my website, appenguins.weebly.com, you can actually find the codes to play any of these games at any time, as well as there's 20 million other resources there. So a plant cell is a 0.0 molar solution, means that the solution is going to be hypotonic to the cell. Since the solution is hypotonic to the cell, the solution is going to move into the cell. And because the cell wall is going to push back against it, causing the cell to be turgid and kind of firm. Nice job, y'all. Which organelle found in a plant cell provides the plant cell with turgor pressure? And I mentioned this earlier in the live, as well as I think I may have mentioned this earlier in the game. So you probably should have the answer to this one. Maybe I shouldn't explain the answer so well, because then you just get the answers to the future questions. I did the biochem a couple weeks, I think 2 weeks ago, or actually last weekend on Saturday, I did the biochem one. So what organelle found in the plant cell provides the plant with turgor pressure? That would be your central vacuole. Central vacuole is going to store that excess water, which is going to then cause that pressure to be pushed up against the cell wall, which gives that turgor pressure. Respiration, we said, is going to be the site of our membrane protein, and our protein synthesis for secretion. Golgi bodies are for modification. Lysosome is for digestion. Yay, everybody got it. Okay, I think we have one more question and then we move on to another game. So what property of the plasma membrane allows for oxygen and carbon dioxide to fuse by simple diffusion? Yeah, I like biochem too, I agree. It's a fun unit. Okay. So phospholipids are going to be amphipathic. The phospholipid itself is amphipathic, but I discovered, I think recently when I was working on some review stuff, that the plasma membrane itself is actually nonpolar. So I actually don't know which answer I picked for this one to be the right answer. I think based on my knowledge now, this one's actually a more correct answer because the phospholipid, technically the membrane is nonpolar, but the phospholipid as a singlet is amphipathic. So again, I wrote all these games, so it's possible that like I don't know how it would be on the actual AP exam. Amphipathic is a true statement for a phospholipid, which is describing for the nonpolar interior to the membrane. But again, there is such a large area that's hydrophobic versus a very small area that's hydrophilic, so the entire membrane is actually technically nonpolar. So sorry, y'all. You can hate me later. Okay, so that's the end of that game. So again, I said that we had two more games. We could take a plasma membrane game, or we can play the organelle game. So it's up to y'all which one you would prefer. I'm going to get rid of this. You said that you have your biochem test. While y'all are deciding which game we're playing, again, type in the chat if you want plasma membrane or organelles. So if you go to apbiopenguins.weebly.com for the person who has their tests, and if you go here to game codes, you'll see all of the games that I have for our Unit 1 stuff, so all that biochem stuff that you can use for your review for your test that you have. And wow, that is so bright because I'm sitting in the dark. Sorry, y'all. Anyway, so do you want plasma membrane or do you want organelles? It looks like we have someone in the chat asking for organelles, so let's play the organelle game at that bright white screen. I shouldn't play these games in the dark. I think it's actually helpful in order for y'all to see the screen though. So our code that we have is 322991. So go ahead and join our game. So 322991. And I would appreciate if someone could type it into the question box so I can pin it so that anybody who may join our live later will be able to join that game. So again, we have 322991 is our code. Thank you for typing it for me. Although you still haven't joined the game, but that's okay. Some point we'll join the game so that y'all can. There, we got somebody. I think it's easier that you don't actually have your names on the stream because then you don't have to try to type in your name. It just automatically gives you a name because that's lots of fun. So give just another moment in case anyone else wants to join the game. I see there's two people that are actually in the room, like in the live stream. But it's possible that they might just be chilling and listening to the questions and kind of answering on their own. So the name generator is funny. Yeah, I agree. We have some interesting names. But I figure that those interesting names are probably better than the interesting names that may come up that could get me banned from doing lives. So I figured that this was our safest alternative. I'd hate to get banned from, doing something fun like this for y'all. Cystic fibrosis is a genetic condition that's associated with defects in the CFTR protein found in the membrane, which organelles synthesize the protein. So what is going to make a protein for the membrane? Okay, so we talked before that our roughy R is responsible for making those membrane proteins. The roughy R is going to be basically studded with ribosomes. Those ribosomes are actually free ribosomes. Once they get a signal, they will then bind to the roughy R to finish synthesizing the protein on the membrane. So depending on the way that signal is and depending on the R proteins, the R groups on that protein, it'll either be a membrane protein and kind of sit in the membrane, or it will be basically put on the inside of the ER to then be excreted or secreted by exocytosis. Golgi bodies, we said, we're going to be for modification. Smooth ER is going to store our calcium and detoxification. Lysosome is for basically intracellular digestion. Fibrosis is a genetic condition associated with defects in the protein. After being synthesized, where does it go to be modified, packaged, and sorted? You should be able to get this answer, I hope. I literally just said the answer. Guess y'all can tell that I made these games like last year. so I don't really sit here and do the games constantly with y'all. I just figure I give y'all access to any of the games. Okay, so after we have a protein that is synthesized in the rough ER, it then goes to the Golgi bodies for that modification. And so that should be our answer, as I said before, detoxification and digestion for the organelles. Yay, our one person got it right. Ooh. So the following data was collected by observing subcellular structures of 3 different types of cells. How can you determine these are eukaryotic cells? So when you look at this, what about this tells you that this is a eukaryotic cell? Oops. Okay, so all cells have ribosomes. Ribosomes will be found in prokaryotic and eukaryotic cells. DNA will also be found in prokaryotic and eukaryotic cells. And so will a plasma membrane that surrounds the cell. So the only thing that's different in terms of a prokaryote versus a eukaryote is that they have membrane-bound organelles. And as you saw in the picture, they have a smooth ER, a rough ER, mitochondria, cilia, and Golgi bodies, which are all going to be considered our organelles. But I guess the cilia could go either way on that because they actually have a flagella, so I wouldn't agree with that one. But other than that, following data synthesized is the same data we saw before. Identify a function of cell X. So when you look at cell X, you see that there is a large amount of mitochondria, and cilia is present, while there's a small amount of smooth ER and rough ER and small amount of Golgi bodies. So you need to figure out what is a function of that cell. I'll never understand TikTok. It says there's no one in the room. Maybe you all left and you're just playing the game on your phones. Who knows? Anyways, so since we have a large amount of mitochondria, it means there's a large amount of ATP being synthesized for the cell. That's a common mistake that students make, is that they think that the ATP is made for the whole organism, but in actuality, that ATP is made for the one cell. So each cell has mitochondria that's producing the ATP for that cell. And then we also see the cilia. Cilia is responsible for movement. So we're probably going to see that this is some type of cell that's going to be moving in some way. So like the cells that make up your trachea, they all have cilia on them that's going to kind of move up the the mucus that has like the debris and random stuff that you inhale. So it can kind of move that mucus up and down in your esophagus to be digested, because we don't really need to be breathing in all that junk. So the answer we have here is mobility, because ATP plus cilia. I'm glad you're still here. I just think TikTok's funny because it shows me that there's no one here. Okay, so we need to figure out what is the function of Y. So Y has a large amount of smooth ER, a large amount of rough ER, a moderate amount of mitochondria. There is no cilia, and there's a large amount of Golgi. So you kind of have to figure out, well, what is the function of Y then? Okay. I don't really know if I allowed it to do multiple options, but so your smooth ER is responsible for detoxification. So detoxification should be an answer because we have that smooth ER. The rough ER is going to allow for the synthesis of proteins. And so, of course, those proteins would then go to the Golgi. So secreting of proteins would also be another good answer. So I think I have both of them selected. Yeah, I do. Both of those are options. Mobility, we didn't really see any cilia. So there really wasn't a logical answer for mobility. And then storage would have to be not really logical because you're really using all of your organelles. So it would be a function of Z. Noticing that Z has, no organelles. So what is the function of Z? Storage should be our answer here because it's not really using any of those resources. So like your red blood cell has no organelles in it. Its main job is that it's going to store that oxygen and then transport that oxygen around your whole body. So red blood cell would be an example of Z. Another thing you could have put for this was that it was a dead cell, so like a cell that doesn't need any of its organelles because the cell is dead. Talk about transport, so like your xylem, all those would be logical answers for this. It was a free response question, and so like I was just picking out some of the answers that were on it. How does location of ribosome affect the type of protein product? Hey, Amy and Mika are here. Hi, Amy and Mika. You should have the game noise even louder so y'all can actually hear the music, which I wonder if you can hear the music on your side. I don't know, whatever. Okay, so when we talk about ribosomes, remember the ribosomes, all of them start out as free ribosomes. Those ribosomes then get a signal which will allow them to become bound. But in this case, this question, free ribosomes are going to make cytosolic proteins, so proteins that are just hanging out in the cytosol, because that's where those ribosomes are. Versus bound ribosomes are going to be attached to a membrane, specifically the rough ER, and so they will make membrane proteins or proteins for secretion. This answer was meant as a distractor. The mitochondria does not require any type of proteins to be synthesized for it, due to the fact that the mitochondria actually has ribosomes themselves, so they do not need they have DNA and they have ribosomes, so they don't actually need the ribosomes from the cell. Remember, they are theorized to be a prokaryotic, basically a prokaryotic cell that was engulfed by a ancestral eukaryotic cell many years ago, and they have a symbiotic relationship. What answer did you pick? Oh, ribosomes. Oh yeah, so you got distracted by the mitochondria part. Where is the mRNA used for synthesis of proteins made? Okay, so the mRNA is made actually in the nucleus. And then after the mRNA is made, it's going to leave the nucleus and bind to a ribosome for that protein synthesis. So remember, the DNA is inside the nucleus. So of course, the RNA has to also be made in the nucleus. Organelle responsible for storing calcium. Ooh, we got shifted. Sorry, y'all. In case you weren't in my live last time, or if you didn't see my photo, I literally have my laptop on top of a clothes basket, and then the my phone is sitting balancing on a pair of high heels on top of 4 boxes of shoes. So I have a great, beautiful setup in a closet here, just so that I can have it nice and dark for y'all to be able to see the screen. Because I discovered when I did last semester that y'all could just see my reflection. It was really awkward. Anyway, so storing calcium is going to be in our smoothie R. It also is for detoxification, and then we also make some of our plasma membrane there. Lysosome is lysosome is digestion. Rough ER is protein synthesis for membranes, and Golgi is going to be for packaging and modification. Hey, it's our last question. Tay-Sachs results from a buildup of fats due to an error in which organelle that's responsible for digesting macromolecules. Okay, so we said that digestion was going to take place in the lysosome. So there's actually an error that's taking place in the lysosome that it can't break down those fats, which causes that buildup. Smooth ER is detoxification, rough ER is our membrane protein synthesis, and then Golgi is packaging and modification. And that should be our last question. Done. Okay, so I have one more game, but it's 4:55. Do you want to play the last game of Plasma Membrane, or do you want to just call it a day and log off? It truthfully does not matter to me, either way. So do you want to play the last game, or do you want to say bye-bye? It's up to you. Game, no game? Game, no game? What do you want to do? Game, no game? No one is answering me. The game is on Plasma Membrane if we're going to play it. Do we want to play it? No one is answering me. I think it's because of the lag. Don't we love the lag on online stuff? Anybody want to continue the games or not? So should I assume that since no one is answering me, we're done? One more game. Okay. Then let's get to that plasma membrane game. So continue. I'm sorry, that white screen I realize is like super bad. Okay, so the code for the last game is 665780. The game code is 665780. If someone can type that into the question box, so I'll be able to pin it for anybody who comes in later. So again, the code is 665780. 665780. I already told you the code. 665780. 665780 is the code. And if someone will type that into the question box, that will be great. 665780. 665780. 665780. So on and so forth. I have no one in the game and no one typing in the question box. That's okay. Sooner or later, someone will enter the game. Hey, someone's in the game. Can someone type it into the question box for anybody that comes later? Please. The code is 665780. That way I can pin it because I can't pin it myself. That's one disadvantage of TikTok, because on Instagram, I think I can pin like what I type into the chat, but I can't pin what I type into like this chat. Thank you. Okay. So we got the code ready to go. 10 questions. Let's roll. Which of the following describes the main components of the plasma membrane? Main components of the plasma membrane. Okay, so the main components that we have are our proteins and our phospholipids. Those proteins make those membrane proteins for signal transduction, for cell recognition. It allows for membrane transport. There's a lot of functions to those proteins. And then the phospholipid actually makes up the full membrane. And so it makes like this fluid mosaic model of the proteins and the phospholipids kind of intermixed together. I will agree that there is a steroid called cholesterol that sits in the membrane that maintains the fluidity of the membrane, but it's not like a main component of the membrane. Which poem describes the orientation of the plasma membrane? Ooh, we have more people than we have. Okay. So we're going to have hydrophilic heads are going to face the exterior and the interior of the cell. They're made up of phosphates, plus a whole bunch of other stuff. And then you have your fatty acid tails facing the interior of the membrane, which makes it hydrophobic on the interior part. So you actually have a bilayer. So you're going to have the two fatty acid tails on the inside, and then you'll have either fatty like your phosphate heads on the outside. So it makes up that membrane kind of. Why is there three fingers there? It makes up the membrane with three like that. Somebody asked how often I'm going to do these. I do them every other weekend. So I alternate between going YouTube live and going live here in terms of these review sessions. So next weekend I'll be on YouTube on Marco Learning's YouTube channel doing an official like review session with like a PowerPoint and something, a little more fancy than this. And then on the other weekends, I go live here at 4 o'clock again Sundays to do games. But all these games are always available to you if you go to my website. So apbiopenguins.weebly.com, and you can play any of my games at any time. You don't really need to be on the live. The live is just designed that if you want to hear the commentary, the explanations to the questions, or also just want to be in a more group model, I guess, for this. So having materials that can easily diffuse across the membrane are going to be small and nonpolar. That's why you can have oxygen and carbon dioxide passing directly through the membrane without any trouble because they are small as well as they are nonpolar. Nonpolar meaning they have nonpolar covalent bonds in them. Which of the following maintains the fluidity of the plasma membrane? And I did mention this earlier. We'll see if you remember what I said. Okay. So the one that maintains the fluidity of the membrane is going to be your cholesterol. The cholesterol is going to be a steroid molecule with 4 fused rings that will bind to the fatty acid tails. By binding to those tails, it's going to inhibit the tails from compacting when it gets too cold or getting too far apart when it gets too warm. So it acts as a buffer in the temperature, and it maintains that fluidity of the membrane. Ooh, I got y'all on that one. Sorry, y'all. Which following describes the plasma membrane of organisms in a cooler environment? Hey, so the cooler environment means that it's going to be really cold and you're going to find that things are going to condense. So you want to find a way for your fatty acid tails not to be as able to compress and condense. So having unsaturated fatty acids would be best for that because that unsaturated fatty acid means that there's a double bond in there that causes a kink. That kink is going to inhibit that compaction and it's going to, of course, maintain the fluidity in a cooler environment. Ooh, I got y'all on that one. Protein is on the cytoplasmic side of a vesicle, which the following describes the location of the protein after exocytosis. Okay, so if it's on the inside of the vesicle, based on the way that everything works out with exocytosis, is if it's on the inside of the vesicle, which is going to be a little hard for me to map this out, but as it goes to the membrane, it's going to open up and secrete it out. So it'll be kind of on the inside of the vesicle and then diffuse with the membrane, so it should be on the outside of the cell. So it'll be bound to the extracellular side of the plasma membrane. Oh, I am wrong. So the cytoplasmic side of the vesicle. Oh, it's on cytoplasmic. Sorry, y'all. My demonstration. I read it wrong. So cytoplasmic should be on the outside of of my hand. So when I fuse with the membrane, it's going to be still on the inside of the cell. I'm sorry, y'all. I was used to the way I wrote the question, which is usually on the inside of my hand, not the outside of my hand. That's what happens when I just read the question quickly. Sorry, y'all. Which following describes the function of glycolipids found on the plasma membrane? Okay, so when we look at glycolipids, those are going to be responsible for cell-to-cell communication because of the fact that they're going to bind to receptors on other cells. Ooh, I got y'all on that one. Sorry, y'all. Which following describes the difference between a transmembrane protein and a peripheral protein? Okay, so it should be that transmembrane proteins are amphipathic while peripheral proteins are hydrophilic because of the fact that if it's sitting on that membrane and it's not going through the hydrophobic core, it has to be hydrophilic. Whereas their transmembrane proteins go across that membrane, causing them to have a hydrophobic region and a hydrophilic region. Oops, wrong button. Next question. 2 more to go, and then we're logging off. In Fry and Edson's experiment, they labeled plasma membrane proteins of mouse and human cells with different markers. Researchers observed the markers of the hybrid cell, which following describes the hybrid plasma membrane, assuming that the mouse cell was initially on the left side. Where did I come up with this question? Oh my gosh. So, because of the fact that plasma membrane is it's a big membrane, right? It's made up of a lot of different phospholipids, and so these phospholipids are independent of one another. So if you think about a crowd, right? You're at a concert and there's somebody that's holding a beach ball, right? The person holding that beach ball can move throughout that membrane in any way, shape, or form. So if you start a concert and on the left side of the stage, you've got a bunch of people with like pink beach balls. And on the right side of the stage, you see that there's a bunch of people with blue beach balls. By the time that the concert actually starts and gets going, those people have now intermixed together. And so you're going to see kind of randomly dispersion of those blue and pink beach balls. So you would actually see a random distribution of those proteins in our phospholipid. And our last question, and then we're logging off. Which following describes why the plasma membrane is selectively permeable? So why is it able to only allow certain materials to pass through that membrane? Okay, so our phospholipid bilayer is nonpolar, so it's going to inhibit our charged materials and our polar materials from passing through. Um, the proteins that make up that plasma membrane do only bind to specific materials and they only allow passage of certain materials. So it kind of depends on where we were looking at that, but it more or less has to do with that the phospholipid bilayer inhibits those charged materials from passing through. And that gets us to the end. Yay! Okay, so I know there's a lag, so I want to try to make sure that it doesn't just cut off and you don't have your answer. So I'm really just going to kill time. Don't forget to tune in next Sunday. I'll be going live on Marco Learning's YouTube channel so that we will be able to go over Unit 2. I'll have my nice fancy PowerPoint. And don't forget, if you go to apbiopenguins.weebly.com, you can access all of my materials, including my 316-page review guide, which some people swear by. So If you don't know about the review guide, definitely check out the review guide. So, hope you buy penguins are just a success. Bye, y'all.